I'd like to welcome you here this afternoon. It's a real pleasure to welcome Lee Hood back to the University of Washington and to the Department of Computer Science and Engineering for what I think is probably his first public lecture at UW since he left to found the Institute for Systems Biology. Lee has been the highest impact person at UW in my 22 years here by far, and speaking for me and my department, he's really had a tremendous impact on what we do and how we do it and how we think about it in teaching and research and outreach and leadership. It's been just tremendous for us, and we are excited to have him back, and we're excited to have the Institute for Systems Biology sitting next to UW with the opportunity for lots of collaboration. So Lee, thanks for being here.
Thank you, Ed. What I'd like to do today is really two things. One, talk about the enormous revolution that's occurring in biology and medicine today, and do it in the context of this new institute that I've started. This is really quite a pivotal time in biology. We can look back and see that we have been building for the last 10 or 15 years to this point, and it's a real inflection point, because as we look forward, we see that we will be doing biology in a new and very different way. Part of the revolution has come about through a really fascinating convergence with information technology, and that's something that I'll get into at a later point in time. Now, this Institute for Systems Biology is all about biological information and the deciphering of biological information. What I'd like to do is give you a beautiful example of how hierarchical and how historical biological information is. And those features add enormous complexity to the analyses that we hopefully can do in partnership with you. Here is a fly, a common fly called the Drosophila. On your left, you can see the wild type, the ordinary fly with a single pair of wings. And on your right, you see a fly that is genetically identical to this one, but for a single mutation. A single mutation created a second pair of wings. And what that means is there are genes that operate at high levels informationally. Now, that doesn't surprise most computer scientists, I don't think. But what the second feature that is really surprising is this mutational event recreates the historical antecedent of this fly. The ancestor of that fly was, in fact, a four-winged fly. So biological information is hierarchical, and biological information is historical. So the fascinating question then is how do we define biological information? And let me take just a few moments to do that. Biological information of the first type is the information that's encoded in your genes and in your chromosomes. As you know, that's a digital code, a four-letter code. Variation in that code generates information. And one of the most common types of information on your chromosomes are the 100,000 or so genes that are present in humans. Each gene can be expressed in a quantized fashion and creates a related type of coding structure called a messenger RNA. And the messenger RNAs allow for the quantized expression of genes, the expression of individual genes. And this messenger RNA can be fed then into a specialized factory to create the second major type of biological information, which is proteins. So proteins have a much richer language. They have 20 letters in their alphabet. And it is the order of those letters in this protein string that specify how the protein actually folds in 3 dimensions to make a molecular machine. So proteins are the molecular machines, catalyze the chemistry of life, give the body shape and form. And there are 2 major challenges about proteins that are in part experimental and in part challenging computational problems. So the first is the question of how, given a particular order of these letters of the protein alphabet, those letters dictate a precise folding into a 3-dimensional structure. So this is called the protein folding problem. And as I've said, it's part computation and it's part experimental. And we'll talk about how the Genome Project is really going to revolutionize our approach to this whole problem. The second problem is the question of structure function. Given a particular three-dimensional shape, how do we know what that protein does? That's very much as if you were a Martian and came down and looked at a car parked in a garage and tried from first principles to deduce what the car did. Almost certainly, you would fail. You would fail unless you saw that car carrying out, executing its function. And so it is with proteins. The structure function question, again, is in part an interesting computational question and in part a very important experimental question. What is interesting is biology for the last 40 years has focused on these two major types of informational elements, genes and proteins, and they focused on them one at a time. And what I would argue is the future of biology is going to be the analysis of complex systems and biological networks. So here are some subset of the 10 to the 12th neurons that are expressed in the brain with 10 to the 15th connections. And of course, this network gives rise to systems or emergent properties that are called consciousness, memory, and the ability to learn. The critical point is if we were to take one of these nerve cells and study it for 20 years and learn everything it could do, it wouldn't tell us one iota more about the system's properties because they emerge from the operation of the system as a whole. So if we are to do systems analysis, we must have global tools that can look at all of the elements, if not simultaneously, very rapidly to see how the flow of information transpires through the network or through the biological information system. And that's really what this Institute for Systems Biology is really all about. So let me give you then a brief introduction to the Human Genome Project and what I think its lasting impact is going to be. The Genome Project, as you know, is about deciphering human heredity, that is, determining the order of DNA letters in the 24 different chromosomes. What is remarkable about the Human Genome Project is it introduced to biology a completely new type of science. I was at the first meeting in the Human Genome Project back in 1985 when Bob Sinchheimer, who was Chancellor of Santa Cruz, called a meeting of 5 or 6 of us to come for 3 days and explore whether a gift of $35 million he had should be dedicated to an institute to sequence the human genome. And I went partly skeptical and partly out of curiosity, and I came away with the realization that the Human Genome Project was about a dramatic new type of science that was really going to fundamentally revolutionize biology. And it's what I call discovery science. Discovery science is taking an object, in this case the human genome, and it's delineating all of its elements, that is the sequences of the chromosomes, irrespective of any particular questions. Biology up to that time had been very much hypothesis-driven, ask a question, do an experiment, and continue in this manner. And indeed, it was this essence of discovery science that was the very fundamental nature of the vehement objections to the Human Genome Project from 85 to 90. And the arguments were vituperous. What scientists then, what biologists then didn't realize is that discovery science creates an infrastructure that fundamentally changes how hypothesis-driven science is done. And it's been true in every single genome that's been sequenced. The science in that particular organism has absolutely been revolutionized. Now, if you think about the human genome with its 100,000 or so genes, you can argue that it is indeed one of the most fantastic software programs ever created. It was created by natural evolution and selection. And this is a program that specifies information that permits all of us to start out as a single cell, a fertilized egg, and through a very ordered and regulated process to develop into adult organisms with 10 to the 14th cells, where you've played a process of chromosomal choreography. That is, in different cell types, you turn on different subsets of these 100,000 or so genes so they can manifest their very different properties. And of course, the human genome is going to reveal a lot of the secrets of human development as we come to understand more effectively how to study that particular genome. Now, Jim Watson, about 15 years ago, made this now famous statement that we used to think that our fates were determined by the stars. We know now that they are determined by our genes. And of course, one of the interesting questions, a philosophic issue of 3,000 or 4,000 years now, 3,000 years, has been this question of nature and nurture. What I would show you now are the left index fingerprints of two identical twins, 9-year-old girls. And what you can see is those fingerprints are incredibly different from one another. So in spite of the fact they had exactly the same genes, the developmental program for generating fingerprints, either through differing interactions with the environment or through different types of stochastic events, ended up with very different patterns. So the fundamental question we always have to ask when we're talking about any type of trait is, to what extent is it hardwired in our genes, and to what extent is it specified by nurture? And that is a question that we can't answer with any precision at this point in time. Now, the Human Genome Project has proceeded with amazing rapidity. In fact, we will have in first draft the human genome done by the end of this year, which is 5 years earlier than it was projected to be finished. And what that, of course, will give us is a map of the highest degree of resolution, a sequence map where we specify the exact nucleotide sequence of each of the human chromosomes. From that, we can derive a gene map that will give us the structure, the organization, the orientation of genes. And from that, we can also derive a very, very important genetic map. All of us differ on average by 1 letter in 500 from one another, and these variations are called polymorphisms. Most of these polymorphisms make little difference. A few of them make us different one from another, some short, some tall, some fat, and some skinny. But other variations actually predispose to disease. And of course, this is a focus of medicine that we'll return to later. So these genetic maps that pinpoint variation are absolutely critical for the future of predictive and preventive medicine. A really interesting question to ask then, in the context of this introduction, is if we look back 15 years from now, what is the major heritage that the Human Genome Project left us with? And I would argue that there are 3 major ideas. One is this idea of discovery science that we've already talked about. The second is the idea that the human genome has, in a sense, given us a periodic table of the elements of life. So just as the periodic table in the 19th century, which specified the relationships of chemical elements one to another, revolutionized certain aspects of chemistry, so the periodic table of life, in a sense, the parts list of life, will revolutionize human biology. What are these parts? Well, we will be able to define and specify the 100,000 or so genes. We'll be able to identify the sequences that actually regulate those sequences, that turn them on and off at the right time and in the right amplitude. This is called the regulatory code. It's a second type of biological information. It's an area about which we know virtually nothing at this particular point in time. We'll also be able to take the gene sequences, both from human and the other model organisms that have been examined, and deconvolute them into their basic building block components, the so-called motifs. And it is these motifs that will be the key to ascertaining function as well as structure of the protein molecules, because the motifs specify, in many cases, certain kinds of structures, and in other cases, not only structures, but functions as well. So there will be a lexicon of perhaps 1,000 of these motifs that will really revolutionize both the protein folding and the structure function problem. Now, finally, the Human Genome Project gives us access to human variability. The next frontier after straight genomic sequencing is the study of human variability and how that variability correlates with physiology and with disease. And again, that's a topic we'll return to in some detail later on. Now, the final contribution of the Human Genome Project has been that it has catalyzed a series of paradigm changes which have, in a compelling way, led us to this view that the future and frontier of human biology and medicine is in fact going to be systems biology. So let me talk about the kinds of paradigm changes and show you how they lead us to this conclusion. So the first idea is one we've already discussed, the idea that biology is an informational science. But we can add a little more dimension to that idea in that we can stress the hierarchical nature of that information. So in a sense, the primitive element is the gene. That element encodes a protein, and proteins actually have characteristics that go beyond the specifications of the gene itself. For example, proteins must interact with other macromolecular elements in almost all cases. Proteins can be chemically modified to change their biological behavior, and so forth. So as we move up from genes to proteins, we climb a hierarchical level with increasing complexity. And so it is when we move into informational pathways. The proteins can combine together to carry out a particular function, signal transduction, metabolism. And in doing so, they create an entity that has these systems properties that we talked about that can't be predicted from an analysis of any one of the elements alone. And likewise, you interconnect many of these informational pathways together, and you begin getting the network of informational pathways that are the fundamental informational backbone of the operation of individual cells. Now, what is very interesting about this idea of biology and information is the hierarchical levels don't stop with DNA and messenger RNA and protein and even informational pathways and networks, but they go to cells and organs and individuals and populations and finally to ecologies. And I would argue that the tools we develop for each successively lower level, when slightly enlarged, can begin to analyze information at the higher level. So what is the challenge then for the Institute for Systems Biology? It is twofold. One, it is to develop tools of a global nature that can decipher very rapidly information at these different levels. And it is two, to be able to integrate that information cohesively together so that we can build models that will not only predict the structure of the informational pathways, but predict their emergent properties as well. The other, a second paradigm change has to do with these global facilities that we talked about. The idea that we have to develop tools that can analyze DNA or messenger RNA or protein at very large levels. The tools collectively to study genes and messenger RNAs are called genomic tools. The tools to study proteins many at a time are called proteomics tools. And I will, throughout the course of this lecture, give you several examples of these tools. An instrument that we developed back in 1986 in prototype form is the DNA sequencer. It's the instrument that has been used to sequence the human genome. And it was essentially built around the idea that we could color code the 4 bases of DNA strings with 4 different fluorescent dyes, and we could use clever chemistries to actually read out successively the bases by virtue of the colored dyes that each of them actually exhibited. And to give you an idea of how DNA sequencing has changed from that first prototype instrument in 1986, if we look at today's instruments, there's been a 2,000-fold increase in throughput and an enormous decrease in the cost for sequencing. And this will just be the beginning. I suspect we'll get another 2 or 3 orders of magnitude quite easily before we finish. Now, to give you an idea of how revolutionary some of the new tools that are coming in this particular case from a small company in San Francisco called Lynx, this is a company that has learned, one, how to couple 1 million different DNA sequences to 1 million little beads, 2, to fix those beads in a flow cell, and then 3, to simultaneously sequence the DNA fragments on those beads for 20 residues in parallel. So you can do 1 million sequences in parallel for a short stretch. This absolutely revolutionizes how we think about doing many problems. For example, to create something called a physical map of the human genome initially probably cost $300,000 to $400,000 and took at least 10 years to get done. We could, with this technology, do it in a single day and do it actually much better than it was ever done before. So these orders of magnitude transform the opportunities that we actually have in front of us. Now, the genetic marker of the future is going to be this single nucleotide substitution that I talked about earlier. And the reason for that is each of us differs from one another on average by 6 million of these base pairs. Hence, there are many, many genetic markers. Hence, we can get very, very dense genetic maps and easily localize genes that control interesting traits. So the really intriguing question that we run into is if we need to analyze 100,000 genetic markers to get a good map, what kind of tools and techniques are we going to employ for that? And there is a small company in San Diego called Illumina that's using optical fibers, in the end of which, a millimeter fiber, they can put 10,000 wells, and into those 10,000 wells they can put 10,000 beads, to which has been affixed a different 20-mer of DNA, each of which has the ability to detect one of these single nucleotide polymorphisms. And what they've demonstrated is by 3 different assays, they can assay these single nucleotide polymorphisms. And what they've also demonstrated is they can put together arrays of arrays. They can put together 10 of these fibers or 100 of these fibers, or in principle, 1,000 of these fibers. And with this kind of technology, you can think about doing analyses at the rate of 4 million SNPs, single nucleotide polymorphisms a day. So that's more than 3 orders of magnitude faster than any other genetic mapping can be done today. So these are the dramatic frontiers that are being penetrated, and the consequences of these changes are going to be absolutely striking in medicine. So these high throughput tools, these global tools that we've been talking about, don't just relate to genes and proteins, but they relate to machines for separating cells. They relate to means for visualizing these informational pathways operating in cells. They relate to the idea of how you can automate the detection of protein-protein interactions, and on and on. So there are a whole series of global tools that we'll have to develop if we are to move to these successively higher levels of biological information. The next paradigm change that I'll talk about is the idea that if you're to study a biological system, just as if you were to study the car in the driveway and you were a Martian, you have to see it in operation. And of course, the only way you can see biological systems experimentally in operation is through model organisms. So model organisms absolutely become the keys to the kingdom for being able to understand systems biology. Now, the Human Genome Project has designated 4 simple organisms, bacteria and yeast, and a simple roundworm called a nematode, and a more complicated fly. And those genomes have been completely sequenced. And a 5th model organism is the mouse, which rivals our own genomic complexity. Now, what's interesting about the simple model organisms is they have a lot of genes that resemble our genes. For example, the nematode has 19,100 genes, and about 70% of them have counterparts that can be seen by string matching, by similarity analysis in humans. So if you want to study a particular human gene, and if you find it in the nematode, you can then study it in that organism, either to ask what that gene itself does, or to ask what it does in the context of its particular informational pathway. And then you can take those insights back to human genes and learn an enormous amount about those. One of my favorite quotes that specifies a lot about model organisms and why they're important is this now famous quote by Max Delbruck. Any living cell carries with it the experiences of 1 billion years of experimentation by its ancestors. And with the techniques we're talking about here, we have the means to unlock those secrets of the past. And again, I hearken you back to the fly with a single mutation, which recreated an earlier form. It goes without saying that this explosion of information, biological information, is going to require a partnership with computer scientists and applied mathematicians. And it goes without saying that you will be providing us the tools that we will need for dealing with biological information. Acquisition, storage, analysis, modeling, display, distribution, and the whole range. But what I would also contend is that living organisms have had 3.7 billion years to learn how to manipulate their digital strings, and they too have interesting secrets that may give the computer scientists insights into mechanisms of information manipulation and so forth. And that's why some of the areas of biology have, I think, revealed some interesting insights into areas related to computer science. Now, what is certainly true is that the biggest computational challenge that we're going to face, in a sense, is going to be to take a genome, and this is a small genome that has about 1,700 genes. It's Haemophilus, a microorganism. And what we'd like to be able to do is deconvolute those genes into their informational pathways and their informational networks so we could come to understand the logic of life in that organism. And even more important, being able to compare that genome with other genomes to see how the logic of life varies and whether that gives us insight into why things are the way they are. Why do systems operate? What are the constraints and the evolution and change that occurs between differing organisms? And this is a topic that we'll return to at a later point in time. So what this leads us to then is this idea that systems biology is going to be an enormous challenge. And it's here I'd like to take up a discussion of this institute and give you some idea of how it came about, what we're going to do, and really, in some detail, what exactly do I mean by systems biology. When I went to Caltech as an assistant professor in 1970, I decided to spend about 50% of my time on instrumentation and 50% of my time doing biology. And over the next 15 years, we developed the DNA and protein sequencers that are kind of the foundational framework for modern molecular biology. And this idea that technology opens up new vistas and new horizons thus became a center point of our thinking about the future. In 1985, I already pointed out that I got involved in the Human Genome Project and came to realize that discovery science can transform how biology is done, but one of the enormous challenges is going to be to integrate discovery science together with hypothesis-driven science. In 1988, we got an NSF Science and Technology Center for Molecular Biotechnology. This was a wonderful, one of the best grant strategies I think ever deployed by the federal government. It was an 11-year grant. There were competitive renewals along the way, but it gave us $3.5 million of discretionary money, and we were able to venture out into areas that could never have been funded by classic R01s. And we came to realize the importance of discretionary money and its ability to catalyze real changes in science. But at the same time, we came to realize the importance of creating an environment that was cross-disciplinary, that had physicists and chemists and computer scientists and biologists and physicists and the like. And it was in 1992 we moved here, created the Department of Molecular Biotechnology. And I would say to a very great extent, we've been successful in doing that. So successful, in fact, that by 1994, we'd kind of used our space up, and it became obvious to several of us that if we were to move ahead in what was then obviously the challenge of the future, namely systems biology, we'd have to have more space. So we approached McCormick and tried to persuade him that we should get a building together with bioengineering to proceed along this course. And he said, well, you're 11th in line, and it'll take 10 years to get a building. So it was then when we started thinking about the Institute as an entity that could be done in conjunction with the university, but at a different location. We tried 3 years to do that with the university, and I think the bottom line is the Institute, as I envision it, needs a degree of control, of flexibility, and timeliness that just can't be found in a large vertical bureaucracy. And it was in December that I resigned and decided to set the institute up and do things in an independent fashion. And indeed, my colleagues that have helped with this are Rudy Abersol, who is in MBT, is one of the real pioneers in proteomics, and he's developed a spectacular core technology that I'll describe very briefly in just a moment. And Alan Adaran, who's interested in immunology in the innate branch of the immune system. And I'll show you some technologies that we're going to be employing using innate immunity. And they've been absolutely marvelous partners indeed. So what, in fact, is this institute really all about? Well, we have three major objectives. We want to do systems biology, and that's really what I'm going to focus on. We have a lot of new ideas about education, and we're certainly committed to the idea that scientists should transfer knowledge to society. But let me talk about the systems biology aspects. It is critical, if we're to do systems biology, that we have to put in place these high throughput technologies that I've been talking about. So we will be putting in place sequencing facilities and DNA array facilities and genotyping facilities and proteomics facilities and many additional other facilities. And of course, what these different technologies have the capacity to do, quite obviously, is to basically interrogate biological information at all of these different levels: the DNA level, the messenger RNA level, the protein level, and the functional protein level. Almost no protein can operate without some type of modification, and that change creates functional proteins. So how do we go about assessing information at these different levels? And what I'd like to do is tell you about two technologies that were developed by the founding scientist for the Institute and give you some idea of their power. So one is the use of inkjet technology to actually be able to synthesize fragments of DNA, so-called oligonucleotides, on glass or silicon chips in such a manner that in principle you could lay down 100,000 of these fragments, each of which represents a different gene in the human organism. And in principle then, what we could do is use these DNA probes, for example, to interrogate the messenger RNA that's present in a normal cell and in a cancer cell. And the way we do this is we label the normal messenger with a red fluorescent dye and the cancer messenger with a green fluorescent dye, and we actually hybridize both of those simultaneously to these probes, and then we can interrogate each gene, each oligonucleotide probe, to determine what ratio of green and red they have. And when you do that, you get a beautiful specification, in this case, of all the genes that are present in a yeast organism. And if the two messages are coordinately expressed, green and red make yellow, and that's what most of the spots here are yellow. Occasionally you can see red spots, and occasionally you can see green spots that are predominantly expressed in one cell type as opposed to the other. So this ability to do these comparative analyses has turned out to be a powerful technology that has matured relatively rapidly and is being widely used now by many different laboratories. So that's studying messenger RNA. Rudy Abersol has pioneered a completely novel and powerful new technique for studying proteins. The idea is you synthesize a chemical reagent which has at one end a group that can label one of the 20 letters of the amino acid language, have cysteine residues. In the middle, you have a linker that can have either 8 hydrogens or 8 deuteriums, so you get 2 different compounds that differ by 8 atomic mass units in weight from one another. And you have at the other end a tag, a biotin tag, that allows you to pull out all proteins or protein fragments that have this chemical entity affixed to them. Then the idea is really a simple and powerful one. You take a cancer cell with its proteins and you label that, for example, with the light reagent. And you take a normal cell with its proteins and you label all of those proteins with the heavy reagent. You mix the two together and then you fragment those proteins with protein-cutting enzymes, and then you can use an affinity separation just to pull out the peptides that have the appropriate labels joined together. And then you can use an instrument called the mass spec, which can measure precisely the weights of these fragments, and you can do two different kinds of experiments. For a given peptide, you can determine the ratio of light to heavy protein fragment, and that determines how much is present in the cancer cell and how much is present in the normal cell. And then you can take those fragments and you can fragment them still further and put them back in the mass spec, and you can actually determine the order of the letters of the protein language, the so-called protein sequence. And from that, if you know the entire genome sequence, you've identified the gene. So in a sense, this is a protein strategy that's totally akin to the two-color strategy for measuring messenger RNAs from two different cell types. And we're setting up to do this on a very large scale. In fact, this is the only site that has these chemical reagents at this point in time, so we have a unique opportunity to explore this kind of technology. What Rudy, in collaboration with our group, has gone on to do then is to measure the RNA expression in a particular interesting biological system that's been perturbed and compare it with the protein expressions. So when the RNA and protein expressions are in conjunction with one another, the dots, the data points fall on this red diagonal line. And when one or the other of these informational molecules is expressed excessively, they fall off the line. And what we can say is every single point that is off the line has associated with it really interesting biology. So the ability to integrate expressed gene information with expressed protein information is going to be ultimately critical to systems biology. Bringing together scientists, we hope to have half the faculty in the institute be cross-disciplinary to attack the few frontier problems of global technologies is going to be one of our major endeavors. We have a long list of things, all of which we're interested in varying degrees, but we're certainly going to be focusing on computational biology, multi-parameter analyses, microfabrication and nanofabrication, as well as single molecule analyses. And I'll talk about a couple of these in just a moment. But the idea is that the Institute wants to continue to create the global tools of the future that will let us ever more successfully integrate these differing levels of biological information. So let me then move to a more detailed description of exactly what systems biology is. And what I'll do is explain this in the context of a system we're studying to develop the tools. And that system is in yeast. We're studying it in yeast because we know the entire genome, so we can interrogate all the genes and, to a first approximation, many of the proteins. And it's a system that metabolizes galactose to a form from which energy can be readily catalyzed. And it carries a series of 3 enzymatic reactions out. So a part of the system we're interested in are 3 different enzymes. Another part of the system is a molecule that permits galactose to be transported in from the outside to the inside of the yeast organism. And then there are a series of proteins called transcription factors that actually regulate the level of these enzymes. And in fact, when you turn on those pathways, you increase their levels by more than 1,000-fold. So the really interesting question is, can we use the tools that I've talked about here to, from first principles, actually be able to come to understand the system that we've talked about here? To give you an idea about how the regulatory molecules work, here are a series of genes that are present in that pathway, and we think there are about 10 known genes present in that pathway. And this is, GAL1 is the enzyme that catalyzes the first reaction in the pathway. And under ordinary conditions where there's no galactose present in the yeast, 2 other of these proteins sit on a control region and keep the galactose 1 turned off because they don't need that enzyme at that time. However, when galactose comes into the cell, it interacts with a third transcription factor, which can then go over and combine with the other two, and that turns on this particular gene. We know this is how that works, so again, can we, from first principles, actually be able to solve that particular problem? So what we have, Vestin Thorsen and Trey Eidegger, are mutations for all the genes that we think participate in the pathway, and these are mutations that have destroyed the function of the pathway. And what we'll do is look at those mutants in two conditions: with galactose absent, that is, the pathway is not running, and with galactose present, the pathway is running. And we can carry out an analysis simultaneously of all the genes, all the messenger RNAs present in the yeast cell. This is a double knockout, and we can show that the two genes that are knocked out have entirely had their activity destroyed. So this gives you an idea of the RNA data that we'll be analyzing, and we're just beginning to carry out protein studies using that technique I talked about a little bit earlier. So the idea is we take a genetic perturbation and we put it in the midst of one of these two environmental conditions, and then we interrogate the yeast organism with these different perturbations, and we examine what the ratio expressed genes and expressed proteins is going to be for each of those different conditions. What we also are interested in doing is modeling this thing from a theoretical point of view, putting the model parameters in, creating the mathematical model, and that model will predict how the expression patterns under these various states of perturbation will actually occur. And then, of course, the important point is to be able to compare these data to determine a goodness of fit, and that goodness of fit then can be used to design and predict the most effective future perturbation experiments to be put through the biological system, and at the same time you can use those insights to refine your model parameters and the nature of your mathematical model. So what happens is we have a reiterative, continuous type of process, and the important thing is that biologists are key in doing the perturbation experiments, but the physical scientists are key in integrating the information and beginning the modeling, and in developing the algorithms that will let us efficiently choose future parameters for this kind of cyclic kind of process. So the idea that the computer scientists and applied mathematician and theoretical physicist have to be intimately commingled with the biologists to carry out this kind of biology is actually an absolutely a central kind of idea. In a similar vein, then, we can move to more complicated systems like the immune system, an interest of Alan and myself. And the immune system, as I've said, has these two branches, an adaptive immune system where there are receptors that can visualize foreign macromolecular molecules called antigens, and its system's properties are immunity or tolerance, that is, the ability, for example, not to react to your own proteins. And we're going to be using exactly the same systems approaches to understand these systems properties, and they in turn give us insights in how to make better vaccines or how to deal with the whole issue of autoimmunity, that is rheumatoid arthritis and the like. Now, the second branch of the system is innate immune system. This is Alan Adarin's level of expertise. Macrophages can be activated, and when they're activated, they do spectacular things. They are, for example, surveillance cells that are very efficient at killing cancer cells. And the way they kill these cancer cells is by virtue of recognizing an antibody that's seen a different or a foreign determinant on the cancer cell, and then interacting with what is called the Fc domain of the antibody through an Fc receptor, and this interaction activates the macrophage to perform a killing function on the cancer cell. And of course, sometimes cancer cells escape from this normal surveillance mechanism by a variety of different ways. And what we're doing in the Institute is to combine the techniques of genomics to identify molecules that are uniquely present on cancer cells, and then the techniques of proteomics to find those molecules that are present in large concentration on the cell surface. And then we're actually going to make antibodies to these particular kinds of molecules. And together with Alan Adarin, we're going to figure out ways to activate the macrophages so we can use this as an absolutely ubiquitous and general approach to cancer biology. It will apply to any type of cancer, and it is a revolutionary new approach that comes right out of these global technologies that I've talked about here and can be applied, I think, in a really efficient and effective fashion. And finally, let me talk about another area, this area of human variation that we talked about before. And if you think about it, there are two kinds of human variation we're really interested in. One is the variation in our genes, in our DNA, and these are called polymorphisms. And what we will be able to do over the next 10 years is identify probably hundreds, if not thousands, of polymorphisms that will let us write out the future health history of the individual with predictive probabilities about your likelihood of getting cardiovascular disease or cancer or particular immunologic diseases. But what looking at the expression patterns of RNA and protein does at regular intervals of time is it gives us an instantaneous assay of where you are in any particular health or disease process. So it gives us a predictive health status. And what we plan to do in the Institute is actually look at 1,000 or so genes in 5,000 normal and diseased individuals. And we hope that we'll select these genes so that they can give us a window into many cancers, a variety of different cardiovascular and immunologic and metabolic diseases, and indeed some other areas. And this will begin the initiation of this predictive health history. Now, the next step, of course, has to be to develop preventive therapies that can circumvent the limitations of the genes whose deficiencies that you've already identified. At the level of messenger RNA and protein variation, what will happen is we'll be able to actually look at the information expressed in your white blood cells because it actually reflects information not just from the immune system, but from the cardiovascular and the nervous system and many other systems. And for each gene, we can, say, once every 2 or 3 months, actually plot out the normal variation, and what you'll see in some cases, it will go above the normal statistical limits of expression. And in this case, you get into a disease category, and you and your physician will be identified that something has to be done about that. So what this does is an instantaneous predictor of health history. Now, I've simplified enormously an incredible number of fascinating technical questions, from the mathematics of the genetic mapping and the identification of the variations to the database problems that exist in this kind of preventive, predictive medicine of the future. But there are enormous opportunities for computation to occur. So let me then quickly summarize the fact that in the Institute, we want to have virtual collaborations. We want to bring in biologists from all over the world, just as CERN does physicists, and let them operate for a week or a month or 6 months to gather the data that they can then take back and work on. We'll set up workshops and scholars programs. And indeed, we have gathered a cluster of outstanding scientists together that will be participating with us in various aspects of the Institute. They, for the most part, will stay at their own academic institutions, but there will be deep and extensive collaborations. And in the same vein, we began 2 types of industrial collaborations. A limited number with very large corporations that have the resources to take on hard, long-term problems. And one example we've already set up with Monsanto is characterizing crop plants and so forth. And this is a project that has been of the order of about 40 million over a 2.5-year period, to give you an idea of the upper end of the size that we have in mind. We're negotiating a deal with Merck right now, and we're negotiating another deal with Immunex now. And we're talking indeed with all of these companies at various levels about possibilities. We're also very interested in small technology companies that have expertise in particular technological areas. So Illumina has this optical fiber array that I've talked about. ORCID is a specialist in microfluidics and links and highly parallel sequencing. So what we plan to do with collaborations with these companies is bring these technologies in and integrate them into our high throughput facilities, both to benchmark them against the facilities that we have and to actually be able to use them to integrate, again, these various levels of information far more effectively. The unique features of the Institute, I think, really are many, and it points out the importance of being able to raise a large endowment, which I'm spending most of my time doing at this particular point in time. But we hope to coordinate discovery and hypothesis-driven science. We hope to occupy a unique niche between industry and academia. Industry has to make a profit, therefore it can't take on long-term problems. Academia has limitations of resources and a willingness to take risks in certain kinds of areas. Deciphering and integrating these levels of biological information is a major opportunity and challenge. How do we bring all of these different scientists in so we can effectively do this cross-disciplinary science? I think the key there, and we're starting to write a textbook on this, is to view biology as an informational system. And it really revolutionizes how you think about and how you teach biology. The CERN of biology I've mentioned, as I've mentioned the virtual institute and so forth. The idea that we can in the institute not only take on prototyping of complex instruments, we can make them robust and actually integrate them into factory production lines at the same time, something that no other academic institution has ever attempted before. And again, that is if we have sufficient resources to do this. We're going to take on big problems of biology and medicine, and we're certainly interested in interfacing biology and society in a variety of different ways. So let me then say, in summary, I've given you the global overview, and I'm inviting those of you who are interested in interacting with us to come and talk with us about more detail. And the kinds of problems that we can talk about really can be viewed in two ways. So one, we can view it in the context of these different levels of biological information. And each of these present enormously challenging technical problems that we'll need a lot of help with. And for example, if we take just one of these, genomics, there are a whole series of different things that represent computational and algorithmic challenges, as well as graphical display challenges and a variety of other things. So many, many different problems for each of the things that I've listed here. But we can look at this whole question in quite a different way, and we can say that IT and biotech actually has a lot of shared problems. How do we capture data? How do we warehouse it? How do we integrate that data? The systems engineering, how are we going to be able to model it and identify key nodal points? The integration of heterogeneous databases is an enormous challenge in biology. Data mining, pattern analysis, the whole question of scale. How do we deal with information that exists at vastly different scales? The systems modeling, graphical display, many different opportunities, quite obviously. Here is Moore's law that says the number of transistors is going to increase every 18 months from 1970 up into the current time. It's been more or less true, and as you know better than I, that's fueled a lot of the revolution in IT. We have in biology exactly the same revolution going on today, an even steeper increase in sequence information coming from the human and other organisms. And of course, the challenge in biology is how do we convert information actually into knowledge? And I would argue it is through the integration and the visualization of information at these different levels so that we'll be in a position to carry out the modeling as I described in that prototypical example. And every level of this specification and integration of information represents, I think, enormously fascinating computational challenges. We've already talked about all the levels of biological information, and we look forward in the future to collaborating with you on pushing the frontiers forward in deciphering biological information. Thank you.